So once the main body axis has been laid down by the primitive streak, the next process is formation of the neural tube, what will be the spinal cord in the adult. And what happens is that the ectoderm on either side of the midline begins to rise up around the groove in the centre. So again, we're looking at a cross-section across the axis of the body of the embryo. So here we can see the folds beginning to swing up, and we've also marked the cells at the very tip in dark green, and those will be neural crest cells. So as the folds are beginning to approach each other, then the cells at the very tip are the neural crest cells. Gradually the system is beginning to form a tube running the length of the body, hinging about a point in the middle. So the folds will begin to approach each other. Eventually they will meet, and where they meet they will fuse together. And the cells that we've marked as the neural crest cells in dark green are left as a separate, isolated little group of cells. And as we'll see later, these have an extremely interesting developmental history of their own. They will give rise to a whole range of different kinds of tissue. But together, what they've done is to form a fusing tube running the length of the embryo, the neural tube or future spinal cord. Neural crest cells marked here, neural tube marked in the diagram as well. Now looking at that from above, again imagine that we were swimming in the amniotic cavity and looking down at the floor of it. We can see that this process is not quite as simple as the initial diagrams would suggest. In particular, towards the head end, it remains fairly wide and open, and that's where the brain is going to form. And the folds are coming together first round about the middle. They'll meet in the middle, and then they'll begin to, as it were, zip up in either direction, so moving towards the head and moving towards the tail. And in the diagram in the bottom right, you can see that this process is largely complete down towards the tail end, but is still taking place up at the head end where the brain is beginning to form. So the folds coming together, first of all in the midline, and then zipping up towards the head and towards the tail. So fusing, as it were, up towards the head and tail in each direction. And you can also see little blocks of tissue on either side. Now, these are not actually in the ectoderm itself. They're in the underlying mesoderm. So they're little cubes of mesoderm forming on either side of the ectodermally derived neural tube. And you can probably guess what these are going to contribute to. They will contribute to the vertebrae. This is the segmental structure of the vertebrae that form the spine, although, as we'll also see in another lecture, they give rise to a wide variety of other kinds of tissue as well. We've marked here the posterior neural pore. That's just the opening at the tail where fusion is not quite completed, and at the forward end, the developing brain, the anterior neural pore, and that's where the brain itself will begin to form. And again, there is a separate lecture devoted to formation of the brain. So just revising that with the labelling on the diagrams, you can see where the primitive node and primitive streak were, and the folds beginning to rise up on either side, coming together to meet in the midline, and then the fusion extending towards the head and extending towards the tail. And you can already imagine, I'm sure, that this process might fail, it might not be complete at various points, and that will cause a variety of birth defects, which we'll look at a little later within this lecture itself.